Welcome to Introduction to Spectrophotometers. So in Chapter 17 we saw a single beam spectrometer where we have light leaving our source. A narrow wavelength range is then selected by our monochromator. That light then passes through our sample and hits our detector. We first measure the irradiance, which is P0. This is in watts per square meter. We measure that irradiance hitting our detector after passing through the reference cell, which is the solvent blank or reagent blank. And then we measure our irradiance P hitting the detector after passing through the sample cell with the sample in it. And the irradiance P is usually lower than P0. And then the fraction P over P0 is our transmittance T. The absorbance A is equal to log of P0 over P. Note here that this fraction P0 over P is the inverse of the transmittance T, and that means that A is equal to minus log of T. So the absorbance is proportional to analyte concentration. Remember, this is Beer's law, or the Beer-Lambert law. Now, one major problem with a single beam instrument is that you must switch between your reference and sample cells, which becomes impractical when measuring absorbance as a function of time, as might be done to study kinetics. So there's drift over time in the source intensity and in the detector response. And this means that after some period of time, that reference that we have originally is no longer going to be valid. But we can avoid these problems at the expense of additional cost by using a double beam spectrometer, outlined down here. Now, in a double beam spectrometer, light alternates between passing through the reference cell and the sample cell. So we've got our light coming from our light source, it goes through our monochromator, and now we have light sometimes going through our sample cuvette to our detector, and sometimes going through the reference cuvette to our detector. Then there's probably some amplifier and some display. So which cell, the reference or the sample, our light is passing through is selected by a chopper, or a rotating mirror. Now when light passes through the reference cell, then P0 is measured. But when light passes through the sample cell, then P is measured. So over here we get some beam chopper, and that's a rotating mirror, and we put our mirror into the path, and that bounces the light through our reference cell, and then we take the mirror out of the path, and that passes the light through our sample cell, and so on. So which cell is being measured is changing a few times every second, and then our electronic circuitry is comparing P and P0 in order to find T and A. Because the sample and reference cells are compared several times every second, then any changes in source intensity or detector response with time and wavelength are automatically corrected for, because we're always measuring our reference cell, and we're always measuring our sample cell. But because there can be small differences between the light paths for the reference and sample cells, then a baseline is recorded first with a blank in both the reference and the sample cells. And then our baseline is subtracted from our subsequent sample measurements in order to obtain our true sample absorbance. So a more detailed schematic of a double beam UV-Vis spectrophotometer is shown here. So our visible light source, which is here, is a quartz halogen lamp similar to that used in some car headlights. And the ultraviolet light source, which is over here, is a deuterium arc lamp that emits in the 200 to 400 nanometer range. Now, a mirror here selects one of our two sources and then passes the light to grating 1, which is there. Now, grating 1 selects a relatively narrow range of wavelengths to then enter our monochromator. So that light then bounces off of this mirror and goes through the entrance slit to our monochromator. Our monochromator then is selecting a narrower band to pass to our chopper, so the light comes in, bounces off a collimating mirror, goes to grating 2, bounces off another mirror, that's our focusing mirror, goes through our exit slit, hits another mirror, and then leaves the monochromator. So note that everything up here in this dashed line is part of our light source, and everything in this dashed line is part of our monochromator. The light then leaves our monochromator, goes to a couple of mirrors, and then gets to our chopper. And the chopper then either passes the light through or reflects the light down a different path, and then the light goes either to our reference cell or our sample cell. Sample cell is here, the reference cell is there, and then the light goes to perhaps another chopper and onto our detector. In this case, the detector is a photomultiplier tube. Now, a photomultiplier tube creates an electric current that's proportional to the irradiance striking that photomultiplier tube. And here is a research quality spectrophotometer. So this is what it looks like on the outside, and here's what it looks like on the inside. We've got our two light sources up here, a deuterium and tungsten halogen lamp. We've got our mirror that selects which light source we're going to use. That light then passes through the monochromator here, goes to a beam chopper. The light then either goes through our reference cell or through the sample cell, and then it goes to our photomultiplier tube. So the rest of Chapter 19 examines each part of our spectrophotometer in greater detail. And that was Introduction to Spectrophotometers.